Hello everyone. I'm NIMSHA. I'm currently pursuing my Masters in Microbiology here in Central University of Tamil Nadu. I'm here to present about the topic microbial endosymbionts and epiphytes. These two are very important modes of association between microbes and plants. Microorganisms interact closely with plants, animals, and other microbes. Two of the most fascinating relationships of, are of endosymbionts and epiphytes. Endosymbionts live within host cells and tissues in a mutually beneficial or commensal relationship. Epiphytes, in contrast, they live in outer surfaces of the leaves, stem, roots, and contribute to the plant protection and ecological interaction without entering the host tissue. In these interactions, they provide plant nutrition, disease resistance, and adaptation to environmental stress, etc. They can also play a major role in host metabolism, such as nitrogen fixation, vitamin production, stress tolerance, and also influencing the microclimate conditions in the case of epiphytes. Next is endosymbiosis theory and endosymbionts. In this endosymbiosis theory, these provide important evolutionary concepts in biology. It explains about how eukaryotic cells evolved from simple prokaryotic ancestors. According to this theory, organelles such as mitochondria, chloroplasts were originally free-living bacteria which were engulfed by primitive host cells. Over time, these engulfed microbes develop permanent symbiotic relationship providing energy or photosynthetic benefits to the host while receiving protection and nutrients providing energy in return. This process gave rise to the modern eukaryotic cell structure. Microorganisms involved are bacteria, fungi, algae, and protozoa, which live inside the host cells. Next is microbial endosymbionts. Microorganisms such as bacteria, fungi, algae, protozoa that live inside the host cell tissue and forms a relationship like mutualism, commensalism, and maybe parasitism, depending on the nature of the interaction. They play an essential role in nutrient acquisition, growth promotion, defense mechanism, and even evolutionary adaptations. For example, Rhizobium species inhabit in the root nodules of the leguminous plants where they fix atmospheric nitrogen into ammonia, which is a usable form for the plants. In return, bacteria receive the carbohydrates and a protected shelter. Next is types of endosymbionts. Endosymbionts can be categorized into several types based on their relationship and necessity of the host. Mutualistic endosymbionts benefit both partners. Example, Rhizobium bacteria fix nitrogen for legumes while receiving carbon compounds in return. Commensal endosymbionts. They are the organisms which benefit themselves without affecting the host without affecting the host negatively. They can still indirectly support the host by competing against the harmful microbes. Next is obligate endosymbionts. Obligate endosymbionts are essential for the host survival. One of the biggest example is the species Bicinera aphidicola in aphids, which provides essential nutrients that the insect cannot produce. Next is facultative endosymbionts. On the other hand, these are not essential but provide advantages under specific environmental conditions such as extreme temperature or pathogen pressure. Next is the role of endosymbionts. Endosymbionts can perform multiple critical functions that uh, contribute to the host fitness and ecosystem balance. Rhizobium can fix the atmospheric nitrogen into ammonia for the plant uptake. Also, mycorrhizal fungi can enhance the fixation of phosphorus and other micronutrient uptake through the hyphal network system. In defense, endosymbionts produce antimicrobial compounds which can trigger the systemic resistance, helping the host resistance to the pathogen. They also influence the developmental regulation by altering the hormone pathways like auxin and cytokines, leading to the enhanced root and shoot growth. In a broader scale, the endosymbionts have a role in massive evolution, driving the organelle formation and promoting horizontal gene transfer. Next is epiphyte. Epiphytes are organisms that live in the surface of the plants, especially on leaves, roots, and stem. They do not harm the host, but they have the influence on the plant physiology, disease resistance, and environmental interactions. One example is Pseudomonas fluorescens, which colonizes on the leaf surface and protects against the invading pathogen by competing for space and nutrients, and also sometimes by producing antimicrobial substances. Epiphytes has role in processes like surface moisture retention, microclimate regulation, etc. Next is type of epiphytes. Epiphytes can be categorized based on the plant part they colonize and the ecological roles. Philosphere epiphytes inhabit on the leaf surface, and the population of the organism, the bacteria on the organism, can be 10 raised to 6 to 10 raised to 7 cells per centimeter square. These communities can include bacteria, mainly Methylobacter and Pseudomonas, yeast, algae, and fungi. They can influence the leaf microclimate, degrade the cuticle waxes, and affect the gas exchange. Next one, rhizoplane epiphytes. They live on the root surfaces. They, by participating on the nutrient mobilization, mainly for example phosphate solubilization, and also promote the root growth, and also generating the also generating the hormones like indole acetic acid. The common species include Azospirillum, Bacillus, Pseudomonas, etc. Stem and flower epiphytes provide protective functions against pathogen, also influence the floral scents, and affect the pollination behaviors. In aquatic epiphytes, algae and cyanobacteria colonize on the submerged plant parts, influencing the nutrient dynamics and oxygen levels of the aquatic system. The role of epiphytes. Epiphytes perform several ecological roles and protective functions. One of the major role is surface protection. They compete with the pathogen for nutrients and space, secreting antibiotics and siderophores, and form biofilms that physically block pathogen entry. They also modulate the microenvironment by affecting the leaf surface humidity, temperature, gas exchange, and which can influence the plant physiology. This can also degrade the cuticular waxes and impact the stomatal activity and surface permeability. Photosynthetic epiphytes such as algae contribute to the additional carbon fixation, while the decomposition of the organic matter on the plant surface enhances nutrient cycling. Importantly, epiphytic communities serve as environmental indicators because they respond to pollutions rapidly And not only pollution, they respond to climate changes and atmospheric conditions, and so they are used in biomonitoring programs to assess the quality of air, environmental health, etc. Applications. Microbial endosymbionts and epiphytes have so many applications widely across agriculture, biotechnology, environmental management, and medicine. In agriculture, symbiotic bacteria such as Rhizobia and Frankia are used as nitrogen biofertilizers, which reduces the dependence on chemical fertilizers. Epiphytic fungi such as epiphytic bacteria such as Pseudomonas and Bacillus have functions as biocontrol agents protecting against the foliar pathogens, and they also promote the drought and salinity tolerance. In biotechnology, endosymbionts produce valuable enzymes, antibiotics, bioactive metabolites used in pharmaceutical and industries. And also, genetically engineered symbionts can introduce novel metabolic pathways, expanding their utility. For environmental management, endosymbionts contribute to bioremediation and breaking down the pollutants in the plant host, etc. Epiphyte enhances the phytoremediation by increasing the surface area of the pollutant uptake and transformation. In medicine, Wolbachia used is used to control the vector-borne diseases by reducing the mosquito virus transmission. It is a suitable method for vector control also. Next is the advantages. Advantages of using microbial endosymbionts and epiphytes are many. In agriculture, they can be used to promote sustainability by reducing the reliance on the chemical or synthetic fertilizers and pesticides. These can be also used to strengthen the plant resilience against both biotic and abiotic stresses. And by supporting the healthier plants and soils, they improve the crop yield and which is a sustainable and eco-friendly way. Supporting essential ecosystem services. On the ecosystem level, microbes contribute to the carbon sequestration, nutrient cycling, and disease regulation. And another advantage is that they are cost effective. They can be often applied through seed coatings and soil amendments without expensive infrastructure. And next one, the last but not the least, can be the advances in the genomics and the synthetic biology, which can allow the genetic engineering of the symbionts to improve the crop traits or introduce the biological functions. Next comes the challenges. Despite of the benefits, several challenges can limit the widespread use of endosymbionts and epiphytes. Major issue can be host specificity. Many endosymbionts are highly specialized and only form successful association with the certain plant species or cultivars, restricting the broader application. Next one is environmental sensitivity. It depends on the activity of the endosymbionts and epiphytes depends on the factors like temperature, soil, pH, moisture, etc. So it is very sensitive to the corresponding environment. Another challenge is competition with native microbes. Introduced beneficial strains may be outcompeted by indigenous microbial communities, reducing their persistence and impact. Next is additionally there are regulatory barriers also, like particularly genetically modified symbionts which can also slow the deployment. Field consistency also is a concern, like strains that perform well under laboratory or greenhouse conditions can fail when we are using in the variable field environments. The next challenge can be the storage and formulation, which are practical, which has practical difficulties, and some beneficial microbes have shorter shelf lives and require careful handling for maintaining their viability. Next is environmental impacts. Both microbial endosymbionts and epiphytes have positive and risky ways potential also. In positive impact, they enhances the soil health, increase the organic matter, stabilize nutrient cycles, and improves the soil structures and fertility also. And they reduces the need of chemical fertilizers and pesticides. They decrease the greenhouse gas emission, chemical runoff, and supporting the cleaner air and water, and also promote the biodiversity by enriching plant associated microbiomes, and also support the beneficial interactions. When we talk about the potential risks which are to be managed carefully, introducing non-native microbial strains, that is introduction of the non-native microbial strains can disrupt or alter the existing microbial community and alter the ecosystem dynamics, and it can also potentially displace the indigenous species also. There is a possibility of horizontal gene transfer to unintended host, raising ecological and biosafety concerns. And also, over reliance on the genetically engineered symbionts might reduce the natural microbial diversity over time, making the ecosystem more vulnerable to the disturbances. For When we conclude, we talk about the microbial symbionts, endosymbionts, and epiphytes, which are vital, which play a vital role in ecology, and they have vital diverse functions ranging from nutrient acquisition to evolutionary processes. They have applications in agriculture, biotechnology, environmental management, and also serve as an eco-friendly alternative to conventional practices. However, successful implementation of these requires addressing challenges also, such as host specificity, regulations, etc. Advances in the genomics, synthetic biology, genetic engineering, formulation technologies are more reliable and targeted in the targeted use of microbes. Responsible use can lead to the sustainable ecosystem, which reduces the dependence on the chemicals, and also climate resilience. These are the references. Thank you.